Hello dear students, I hope you all are fine. So here your teacher Raminder Kaur and this is our chapter number 5, Principles of Inheritance and Variation. So this is our fourth video lecture and today we will discuss about the topic Chromosomal Theory of Inheritance. As we have already studied, Gregor John Mendel, who is known as father of genetics, he worked upon genes which he called as factors or units. So three laws we have studied under that: law of dominance, law of segregation and law of independent assortment. During that time when he was working and performing various experiments on garden pea, Pisum sativum, because at that time communication was so difficult and there were other many problems which came into existence, so he was unable to make a proper recognition of his work to his colleagues and other members. So he published his work in 1865, but for several reasons he was unable to make the recognition of his work till 1900. So let's talk about what were the reasons behind this non-recognition of his work, because this is very important question. As we have already talked, at that moment communication was not so easy and it was not quick, right? So he was unable to commercialize his work, he was unable to explore his work to the other colleagues and the whole world. The number second reason was he talked about genes and pair of alleles. So this information, it was not accepted by the other colleagues who were also working in the same field. Number third, that he took the approach of mathematics along with the science. So many of the scientists, they were not recognizing his work because they were not able to understand this kind of approach to make sure that how the biological work or the biological phenomena and laws can be explained with this way, where he took genotypic ratios, phenotypic ratios, Punnett square, means mathematics was used. And the last point was, as he took out that genes are the only discrete units which are passing on from one generation to the another or from the parents to the offsprings, he was unable to prove it to the world because at that moment very simple microscopes were there. Now let's talk about when his work was published and a recognition has been started. So it was a time during the 19th century. Three scientists: number one, De Vries. So generally you almost know, like in evolution also we have studied, it was Hugo De Vries. Then Correns, and third, von Schermack. They were not working together, but they were working separately, and they have rediscovered. Actually what they were doing, they were analyzing, they were taking up, evaluating an analysis of the work done by Mendel on the inheritance of characters. So at that time, because microscope advancement was also there, from simple and primitive microscopes, now there were the presence of electron microscopes or compound microscopes, right? So it was easy to understand the procedure of the cell division, that how cells are dividing into the daughter cells, how mitosis and meiosis takes place. So even at that moment they started to understand that there are some structures which are present in the nucleus which are able to carry something which is passing on from one generation to another, that we today call it as chromosomes, right? So during 1902 they started to understand the chromosomal movement, that how chromosomes, once they segregate and then they are passing to the next cells, like if there is one cell, it will divide and it will make into two cells, two cells will divide into four, so this is meiosis, right? When the diploid cell converts into haploid cell. So here what happens, the segregation of the chromosomes takes place. Now The scientists, two scientists which came and who explained this whole theory of chromosome inheritance, their name was Walter Sutton and Theodore Boveri. Usually we call them as Sutton and Boveri. What they analyzed? They studied that the chromosomes function and the gene function is actually similar. They thought that chromosomes and genes, they are functioning parallelly, their behavior is almost same. So then they were able to explain the Mendel's laws, all the three laws. Now, if we talk about mitosis, that is equational division, and meiosis, that is reductional division. So very important thing here to learn is that both chromosomes as well as genes, they are occurring in pairs, right? So whenever two alleles of a gene pair, they are located on homologous sites. What are homologous sites? Let's suppose there are two chromosomes such as X and X. So these XX chromosomes, they are present in female. XY chromosomes are present in female, male. So in this case, when the genes are located on the same site of the two chromosomes, genes are always in the pairs, right? So when two genes, they are present on the same site of the homologous chromosomes, this is known as sister chromatids. The chromatids are those thread-like structures of the chromosomes, and if genes are located on the same location of both the chromosome, they are called sister chromatids, and chromosomes are called as homologous in nature. In figure 5.8, here they are explaining that how during the meiosis, that is the reductional division, how the germ cells will be formed, and how from the chromosomes which were already present at the very first stage. So G1 and G2 phases we have already done, that how they are at the beginning stage, because chromosomes have to duplicate before the cell division process. So this is the phase when the cell is keeping itself ready for the cell division process. So meiosis 1 and meiosis 2, it is taking place at two stages. So there are four different phases under each meiosis, like prophase 1, then metaphase 1, anaphase 1, telophase 1. Similarly under meiosis 2 also, prophase 2, metaphase 2, anaphase 2 and telophase 2. So during anaphase, it is a very prominent phase when the chromosomes, they will segregate, because now two different cells will form. From two different cells, four different germ cells will form, and all the chromosomes, they are now segregated. From one cell, four cells have been formed, and the chromosomes, they have segregated. Let's talk about what is very much common and what are the dissimilarities among chromosomes and the genes. Here you can see there is one table given and three differences, or you can say three similarities are given. If you look upon under A and under B, all 3 points are similar. Under A, number first point occur in pairs. Under B also it is occurring in pairs. Number second, whenever gametes will form, there will be segregation. Okay. Number third, that when they will segregate, the segregation will be independent from each other. Now, can you recognize either A is chromosome or B is genes? Or you can say either A is genes or B is chromosomes. How will we decide? It means that chromosomes and genes, they have similarities and they are functioning parallel to each other. Now let's talk about during the anaphase of meiosis 1. You can even look over this figure 5.9, that is independent assortment of chromosomes. According to Mendel, he talked about independent assortment of genes. So how they are functioning parallelly, what are the basic similarities among both of them is that during the anaphase, two chromosomes, their pairs can align at the metaphase plate, because there are two opposite sides, two poles we can say, and they are independent of each other. Like in this case, if we talk about the colors, they took four different colors to explain the type of chromosomes. If you look upon the meiosis 1 anaphase, what they have shown in the left column, there is one orange and one green color chromosome is there. And if you look upon the right hand column, what is a possibility? How will they segregate? orange chromosome is segregating with the red chromosome. So it depends. Means orange and short green chromosomes and yellow and short red chromosome. They are at the same pole in the first one left hand column, and on the right hand column there is orange and red small one, then yellow and green small one. They are at the same pole. So in this case, during the anaphase of meiosis 1, when the stage will pass on to anaphase of meiosis 2, what will happen? How they are segregating? So this is very much simple, that if orange is present opposite to green, so they will segregate together. If yellow is present opposite to red, they will segregate together. So same you can see on the right hand side column also. Orange has segregated with the red and yellow has segregated with the green. Apart, there is independence, because orange doesn't mean that it will always go with green. So they are very much independent. So similarly, as Mendel talked about, the genes are always sorting out independently regardless of each other colors or each other characters, whether they are dominant or they are recessive. Similarly, chromosomes are also independently separating from each other. Now, so Sutton and Boveri, so they argued that during the pairing, during the separation of the chromosome, similarly the genes also segregate. We cannot say that only chromosomes are segregating. During that, genes or factors or those discrete units, they will also segregate from one another because they are lying on the chromosome. So if we talk about the Mendel principles, and if we talk about all the knowledge gathered by the Sutton, that's why this theory is now known as chromosomal theory of inheritance. I hope the Sutton and Boveri experiments are clear to you. Now let's talk about if Mendel took the sample pea plant, because it was having various advantages to take over the pea plant. It can undergo its self-pollination, cross-pollination, even the reproduction was very easy. There were 7 contrasting characters available, easy to grow, right? So now Thomas Hunt Morgan, who took over the chromosomal theory of inheritance and performed various experiments in animals. So here was the very first insect that has been taken is fruit flies or vinegar flies, and its scientific name is Drosophila melanogaster. Why they were so suitable? Why not any other insect or on any other animal? These kind of experiments were not performed. So let's talk about that, why it was easy to study these kind of flies. Number one, they can be easily grown on simple synthetic medium in the laboratory. So there was no need to have some complex materials. Number second, their life cycle is very small. It is only 2 weeks, and with the single mating they can produce large number of progeny. So it was easy to study the inheritance of characters from one generation to another. Number third, like you can easily differentiate in figure 5.10 that male and the female, they look different, not only in their size but even there are certain other distinguishable features. And number fourth, there are many types of heredity variations that can be seen with the low power microscope rather than of having the advanced technology. So this was regarding the chromosomal theory of inheritance, which took forward the concepts explained by Mendel, and the chromosomes came into existence. Thank you students. In next video lecture, we will talk about linkage and recombination.